This is an up-close look at figures about the Lac Operon from Weaver's Molecular Biology, the textbook for MCDB 427 at the University of Michigan. My name is Christina, and we'll be going over the Lac Operon today. The Lac Operon is a series of genes found in E. coli which molecular biologists use to better understand regulation of transcription in cells. The Lac Operon is used by E. coli to break lactose down into glucose so that it can be used for energy in the cell. We will be focusing on two things in the Lac Operon, the Lac I, which is right here, and the lacO, which is right here, and it's a cis-acting site. The Lac I gene makes the repressor protein, which is also called Lac I, which is shown right here, which then binds to the operator sequence lacO. When Lac I does not bind to lacO, RNA polymerase, or RNAP, will bind to the promoter sequence, which is right next to the lacO, right here. It will then transcribe the following DNA needed for lactose degranation. So one of the figures looks at the effects of IPTG. So let me just spell that out for you guys so you know what I'm talking about. It's IPTG. And IPTG stands for isopropyl beta D1 phthalogalactoperoxide. And this is an analog of lactose. So when IPTG is present, Lac I thinks that lactose is present, and therefore it will not bind to lacO, and transcription can occur. So I'll show you what it does. So it binds to Lac I somewhere, like this. And then Lac I will not bind to this lacO right here. So then RNAP can then bind to the promoter region and continue to transcribe. Now, in order to measure what was happening to transcription, the researchers labeled the DNA with gamma-labeled phosphorus, or 32P. So I'll show you how they did that. So they had phosphates. This is their DNA. An A sequence, and then another phosphate, and let's say another A, and another phosphate, and so on. This keeps going. So you're unlabeling your DNA, so you're looking at this phosphate right here, and this is now a gamma phosphate. When you unlabel, you make that a gamma phosphate. And then they added increasing levels of Lac repressor. Then the repressor that they used was Lac I. So then they ran two experiments, one that had IPTG and one that they did not. So then they filtered an assay, nitrocellulose filter binding assay. So let me show you what that actually looks like. So here's what it looks like right here. This is nitrocellulose filter binding assay. So in this first picture right here, you have double-stranded DNA. Now this is all radiolabeled with 32P. So when it's passed through the filter, nothing sticks. It all goes into the filtrate. However, when you have protein, the protein sticks to your filter, so nothing will be in your filtrate. Now when you have protein and DNA complex together, your protein sticks to your DNA. Well, some protein sticks to the DNA, or other way around, the DNA will stick to the protein. It doesn't really matter. But this is labeled with 32-phosphate, your DNA is. And your protein will all stick to this filter right here. So now what's not shown is there could be some double-stranded DNA that is not bound to your protein, and that would be in the filtrate. And they don't show this because they just want to emphasize what DNA-protein complex does. So now here we can look at this that's on the filter, and we can see how much emits this radioactivity, and which is how we can tell how much is bound to the protein. Now there are limitations to this, one of which is that you have to have double-stranded DNA. You can't have single-stranded DNA. And that's okay for us because we do have double-stranded DNA. Another thing I want to mention is that our protein that we're working with is your Lac I, and that our DNA, again, is labeled with 32P. And that emits that radioactivity, which we can record. So here's the first experiment that they did. This graph shows that the amount of DNA that is bound to a particular concentration of repressor. And we know that because the x-axis is your repressor concentration and your y is your percent repressor bound to your DNA. And they ran two experiments, one without IPTG, which is this red line right here, and one with IPTG, which is this blue line right here. Now there's a few conclusions we can make by looking at this graph. So first, I want to point out that there's background noise, and that's right here. Because when you have zero repressor concentration that was added, you still have some repressor that's bound to DNA. Without IPTG present, you have 33% that it levels off at around 33%, so you know that that's about how much maximal you'll get bound to your DNA. You also can see that with IPTG present, there's a minimal increase in DNA bound, but not that much. So from this graph, we can see that IPTG represses the repressor function. So the main conclusion that we can make is that when you add IPTG, Lac I can't bind to the lacO sequence. So if it can't bind, then transcription occurs, but you can't tell that from this graph. All you can tell is that Lac I cannot bind to lacO. You don't know the mechanism behind it. Even though I told you what the mechanism was in the first figure, you don't know the mechanism behind it by just looking at this graph alone. Now here's another figure that we're going to look at. So this looks at the effectiveness of Lac I binding to the wild type lacO versus a mutant. The experiment was run as we had previously run the other one, which is on nitrocellulose filter binding paper. However, this was done in the absence of IPTG because, as we saw in the previous graph, when you have IPTG, you will have repressor that is bound to DNA. So we didn't want to run it on something that wasn't going to work, which would be with IPTG. So there's 3 different experimental groups on here, which is your wild type lacO, which is your O+, right here, this red line right here. Then you also have your OC, which is your constituent mutant, which means that the operator mutant is constantly bound to RNAP, so your transcription is always occurring. Now you also have this line right here without an operator, so you don't have a lacO on the sequence. Again, we have our x-axis and our y-axis the same, your x being the repressor concentration with your y being the percent of repressor bound to DNA. Now there's a few conclusions we can make about this graph as well. So we see that the wild type levels off at around 15%, and we also can see that the constitutive mutant levels off at around 20%. When you increase the amount of repressor added with no operator being used, there was still some DNA that gets bound. So that's shown right here. So lacOC is a mutation that Lac I cannot bind to the O. When there is no operator, Lac I shouldn't bind, and it shows that there's no binding, so this is our control. So by looking at this, we can say our control is working. And the main conclusion we can make from this is that lacO is the binding site for the repressor. Thank you guys, I hope you enjoyed, and thank you for watching.